Um, so today we are going to talk a little bit more about some aspects of MHC and some details of MHC genes. I don't know why the Zoom thing is showing up in front of the title, but maybe. The problem is that I have so many things open right now because I have a screen recorder and I have Zoom that who knows where my cursor is. So I think we're just going to have to live with it because I just don't know. All right. So I told you guys last time about the diversity in HLA. So the fact that the MHC is the most diverse gene in the human genome. And before I get into the general structure function stuff, I wanted to give you a couple of fun stories about what happens if MHC is not diverse. Because there are a couple of animals that famously have not diverse MHC. And so I just wanted to tell you a little bit about them. One of those is actually the cheetah. So you may have heard about cheetahs being endangered and a lot of issues with cheetahs. Basically, what seems to have happened with the cheetah population in the past is that the cheetah population went through a genetic bottleneck. The population dramatically shrunk. So there were very few cheetahs to make all the rest of the cheetahs. As a result, all cheetahs are really closely genetically related to each other, and all cheetahs basically have the same MHC. And so they don't have diverse MHC. And so the worry is that cheetahs actually could have a microbe that could sweep their population and could, in fact, kill the entire population because they are so inbred and they don't have MHC diversity. And so, in fact, if you go to South Africa and go to cheetah rescue and go pet cheetahs, you have to actually go through a whole process of sterilizing your hands and feet and all sorts of things so you can pet the cheetah, like I pet the cheetah. And all of this is actually, and cheetahs are sort of the famous example of what happens if something isn't genetically diverse. But in fact, the cheetah's problem is that their MHCs are not diverse because of that genetic bottleneck. So that's one of our two organisms that's an example of what happens if MHC is not diverse. The other example is an organism known as the Tasmanian devil. The Tasmanian devil lives on the island of Tasmania, near Australia. You can see a picture of the Tasmanian devil here from your textbook. And I both have to tell you a few things about Tasmanian devils' lives, and then I'm going to tell you one other thing that's going to feel like an aside, but it's not. So if you look at this picture of your textbook, one reason why I really like this picture of the Tasmanian devil from your textbook is because it shows that it looks like it's going to bite something. And if you read, say, the Wikipedia page about Tasmanian devils or National Geographic page or anything about Tasmanian devils, you will see biting as a major part of their lives. The strength of their bite is actually really noteworthy, and like biting is how they interact with the world. They just bite each other to like communicate. They just bite things. Like biting is a big part of their life. So I like the fact that your textbook shows one like it's going to be biting. Remember that. We'll come back to that. So there's one other thing that we kind of have to think about. So with MHC, one of the things I mentioned to you last time was that matching different MHCs is a big part of transplantation immunology. If we try to transplant an organ from one person to another and the MHCs don't match, the organ is rejected. So there's basically an immune response that kills the organ. So if, for example, I decided to give Sebastian my kidney right now, I'm making the assumption that Sebastian and I have different MHCs, though I have no idea if that's actually true or not. But we'll make the assumption that's the case. If I just tried to give Sebastian my kidney, Sebastian would reject my kidney because we have different MHCs. The reason why that's important is that let's imagine for a second that I have skin cancer on my hand. And I came over and I wiped it on Sebastian. I know, it's a good time. Would Sebastian get skin cancer? No, why not? Yeah, Rishi.
Well, okay. So first, cancer is not like translated.
Okay.
I mean, through like person to person.
Okay.
Also, there has to be mutation.
Okay, so all of that is true, but we need to actually think about why we're not thinking of cancer transmitted person to person. Because we need to think about what stops that cancer from being transmitted person to person in that case. Yeah, Sydney. Yeah, so in fact, if I wiped my skin cancer on Sebastian, the reason why it would not transmit person to person is because his immune system would reject it. In the same way it would reject a kidney, because our MHCs don't match. So that's actually why cancer isn't transmissible person to person. So if I have a mutated cell, that's why I don't transmit it to you, because your MHC would reject it. Unless, of course, we were Tasmanian devils. Because Tasmanian devils also have gone through a massive genetic bottleneck, and basically Tasmanian devils are almost entirely genetically identical within the population. There are actually relatively few of them in Tasmania, and the Tasmanian devil is getting close to extinction because of something known as the Tasmanian devil transmissible facial tumor. So in fact, in Tasmanian devils, cancer does transmit from devil to devil. There's this face tumor. They bite each other in the face. They rub their faces on each other, and they actually spread this tumor. And it is leading to massive population declines because they have no MHC diversity, so they actually can't reject this tumor. There are a few, I think we're now at 4 known transmissible tumors. 2 of the 4 are in Tasmanian devils because they are so genetically inbred. There is also a genital tumor in dogs, and there's also a clam tumor that seem to transmit from organism to organism. And really that's all because of weird MHC-ness. So that's the other thing that's really good for us about having diverse MHC is that we don't, in fact, as Rishi said, we don't have cancer that's transmissible from person to person because we can reject it. Here, where their MHCs are so close, they actually cannot reject it, and they may be going extinct. And so that's my other piece to this. Yep, Jen.
I was just curious if this was like something that happened naturally, or
So basically it was a mutation that happened in one devil, but because they're so inbred, it is actually a transmissible tumor. And it's a face tumor, and their life is about biting each other in the face. So that's that. And yeah, this is another figure from your textbook, because this is a pretty famous story among immunologists. All right, so now we're going to switch gears a little bit, and we're going to be talking about some aspects of MHC genetics, particularly some nomenclature surrounding MHC genetics, as well as then we'll get into some details of the protein structure. But first we're going to talk about the genes, then we'll do the proteins. I've already shown you this image last time, where we talked about the HLA locus, which is the MHC locus in people. And if you remember, I told you about 3 class 1 genes, A, B, and C, and 3 class 2 genes, DP, DQ, and DR. And I told you that these are the most polymorphic genes in humans. So we all have different versions of them. So it is possible, again, I am just completely making this up, but I'm doing it like this way for a reason. It's possible that Karen has HLA-A1 and HLA-A2. And Sydney has HLA-A3 and HLA-A4. And Sebastian has HLA-A5 and HLA-A6, and so on. So this is how we name the different alleles in people, is we put a number on it. Sometimes there are some that are named things like A01, like 27, like subtypes. We're not going to really get stressed about that. Just know that we name them with a number. So like the different variants are done by different numbers. So that's pretty straightforward. Unfortunately, the mouse system was discovered separately from the human system, and people didn't know they were looking at the same genes and the same proteins for a while. And as a result, the way we do the naming for mice is a little bit different. So these are the types of experiments that were used in order to map the mouse MHC genes. So mouse MHC genetics were all figured out by doing skin grafts between mice. This is one of the types of experiments that was used. So here you can see that we have a mouse. Right now you can imagine it's orange. We'll get into the nomenclature going on. You can see it has 2 orange chromosomes. Officially, they're both labeled as B here. And we have this yellow mouse, has 2 yellow chromosomes. And we have a progeny. The progeny has 1 orange, because that's all this parent could have given it. That's the only choices we had. And we have 1 yellow. That's the only choice we had. So we can see our inbred strains, the B strain that's orange, the K strain that's yellow, and an F1 between those 2 strains. And so these were sort of the mice, or the types of mice that were used in these experiments. Please note in this experiment was done with inbred mice. Sometimes people see the next slide about the details of like the transplants and take home with it like who in their family they can get kidneys from. You are not inbred. It doesn't work this easily because you are not an inbred mouse. So don't just take this and like call your family members and be like, what's up? You're going to give me some skin or a kidney. Because it's not quite as easy in people. And so what was seen here was that you could take one of the parents, so you could take this B parent, and you could do skin grafting to another B mouse. Now basically those mice are genetically identical, so it's not exactly a shocker that you can get a transplant to work. And if you tried to do a B transplant to the other parent, the genetically different parent, the K parent, it didn't work. You could also take a skin graft from that B parent and give it to the progeny, who is partially B. See, they're partially orange. And the transplant works. You can do the same thing with the other parent. So our K parent can transplant to another K mouse. Our K parent cannot transplant to a B mouse. And our K parent can transplant to a BKF1, because this one also has some orange or some yellow in it. But if we try to take one of these BK progeny, that's half B, half K, it's half orange, half yellow, we cannot transplant into either the B or the K mouse, because in fact, there's some non-self in either case. This one, the yellow is non-self. This one, the orange is non-self. And so we can only go from the progeny, but we can go from progeny to progeny, all of whom are mixed, B and K. And so we can't go from one parent to another. We can go from parent to child. We cannot go from child to parent. And so, yeah, you, like I said, don't imagine like your mom's going to give you kidneys because of this. Basically, a lot of genetically different mice were used in crosses like this to try to figure out the genetics of MHC. And the mice were really named things like B and K, like this. As a result, the mouse MHC locus was named the H2 locus. So just like we call the human MHC HLA, so I would call them HLA-A1 and HLA-A2, we call the mouse MHC locus H2. The mouse MHC locus has 3 class 1 genes called K, D, and L. This figure from your textbook shows correctly that K is sort of far away from the other two. I always draw them together. I don't care if you you can always draw them together. We're not going to stress out about the fact that K is far away. So I always just put K, D, and L together in the class 1 region. And then we have one that's known as IA and one that's known as IE. They're listed here as A and E. And those are the class 2s. And so just like with human, the class 1s have 1 letter, K, D, or L. The class 2s have 2 letters, IA or IE. So that's good. And so this is the equivalent of the A, B, and C. And so in human, I would say that the human MHC class 1s are HLA-A, HLA-B, and HLA-C. And I would say that the mouse class 1s are H2K, H2D, and H2L. So that's me just listing the names of class 1s. So you can see I've got this thing that tells me what species and then which particular gene we're talking about. The place where everyone gets baffled with the mouse nomenclature is how we deal with different variants. So I told you a few minutes ago that with human, we talk about HLA-A1 versus HLA-A2 versus HLA-A3 versus HLA-A4. We use numbers, and that's sort of sensible. We don't use numbers for the mice. We do it in a whole other just special way. And we do that based on knowing some things about different mouse strains. So the 2 most famous mouse strains used by immunologists are either some that are called BELB-Cs, which are up in the upper left, or C57 black 6s. Those are the ones we used in lab, and they're in the upper right. And someone just decided, so both of these mice are inbred. They're inbred strains. What that means is that they have 2 copies of identical chromosomes. So this mouse, for example, right now we're going to do the BELB-C, has these 2 identical chromosomes. And it has the identical version of IA from both chromosomes. It got the same one from its mom and its dad, because they're identical genetically too. And so we decided to give a name to that version of IA, that variant of IA. And the name we gave it was IA of D. So it has a superscript D. So a BELB-C mouse has IA of D, IE of D, K of D, D of D, and L of D. So if I talk about something being K of D, I'm telling you that it's the K MHC class 1, and it's the D version. So this is kind of giving you what the number would be. And so this is what we find in a BALB/c mouse. And in fact, we call this whole chromosome from the BALB/c mouse an H2D chromosome, because every single one of these is the D version. So in fact, the D version is named as the same one that BALB/c has. And so if I ever tell you you have a mouse and it's of the H2D type, you should be able to expand it out and say, well, that means its IA is D, and that means its IE is D, and that means its K is D, and that means its D is D, and that means its L is D. So for some people that clicks right away, for some people it doesn't. So I want to give you some sort of other examples of this for those of you for whom this did not click right away. So the black 6 mouse is of the H2B type. So because this mouse is of the H2B type, I know it has all these genes, because every mouse, that's the ones that all the mice have. But if we want to know about which version the mouse has, we have IA of B, IE of B, K of B, D of B, L of B, IA of B again, IE of B again, K of B again, like that. So what would we have, what would be in our mouse if I made a mouse that was a progeny of a BALB/c and a black 6? So if they together had a baby, what am I going to have? Karen? Exactly. So they'd have one of each chromosome. So they'd have so that's what that mouse would have. Okay? So I've got two other examples I'm going to draw out for you. I need this side of the board. So this is what would happen if I just picked a random person. They would have all of these genes, which is what people have, but their DP might be number 1, and their DQ might be number 2, and their DR might be number 3, and their A might be 77, and Their B might be 12. And their C might be 32. And then their other DP could be 4. And I may not have enough colors. I may run out of colors. So maybe their DR is also 4. Their A is 32. Their B is 77. Their C is 2. And their DQ is 3. So you can see how they just have all sorts of different versions of these because they're outbred. They're just whatever ones you got, right? Does that make sense? So then, if I go catch a mouse in my apartment, so it's an outbred mouse, it's really the same thing. Their IA might be of B, their IE might be of D, their K might be of W, their D might be of K, their L might be of S, their IA might be of Q, their IE might be of A. I'm out of letters again, or colors again. So K gets to be D. This one gets to be W. This one gets to be B. So those superscript letters are serving the same purpose as the numbers in terms of just telling us which version they are. The only difference is that it's not like there was a person that was number 1, and we decided all of your things get to be called the number 1. Whereas we did decide in this mouse, all of its stuff got to be the Q. And in Blexix's, all of them got to be the B's. And in Balb's, all of them got to be the D's. That's all that means. So don't freak out by the nomenclature. You will see some of this nomenclature on practice problems and things like that. There's a practice problem due the Friday after break, and it will show up on Moodle today. So you'll be able to see and use all of the stuff. In fact, in theory, you should know all of the stuff for it by the end of today. And because we have this, this entire chromosome that all contains Ds is known as the H2D locus. The H2D like this. You can't make up a H2 whatever for this mouse because its stuff is all crazy. Yeah, Addie.
So, what I'm confused about is that there's a finite number of letters, but not the numbers. So, how do they only have 26 possibilities with the mice?
I think they have not cloned out that many of them, but I don't know all of the options. So I do not know the answer to that. It looks like here they have a couple of them where it says H4, H2. So perhaps they've done that, but honestly, most of my life I have used either B's, D's, or K's. So I don't know what the fancy ones would be, but I guess that you do it that way. You will never have to worry about that, but good question. All right, so that was kind of our general discussion of MHC genetics. We are now going to switch gears to thinking about MHC function, most of which we're going to be thinking about kind of the protein that is encoded by the genes we just talked about, and kind of some of the details of that protein. And so right now, we're largely going to focus on the structure of this MHC molecule, and as well as sort of the peptide it has in its peptide binding cleft, and some specifics of the nature of that peptide. We will make a couple of comments about the peptide that gets onto that MHC, although more detail of that is what we're doing next week. In getting into this discussion, there's one other thing I need to remind you of briefly from Bio-250. And that is about the makeup of proteins. So sort of glad Dunaway just walked by and isn't walking by now, because he would be like, they know this, why are you telling it to them? So remember that proteins are made of amino acids. So you can see two amino acids joining together here with the OH from one and H from another coming off to make water. And you can see we make this nice peptide bond. And so what you can notice is that we've got sort of this chain. You can see it better in this little protein at the bottom, where you have sort of NCC, 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 NCC. You've got this whole big long chain. I'm going to refer to that as the main chain, or the backbone. Okay? So if I mention the main chain atoms, or the backbone, I mean this string of NCC, 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 NCC. And what you can notice is that no matter what amino acid we're talking about, all the backbone is identical. The place where different amino acids differ is in this thing called the R group. And so different amino acids have different things at this R place. Everything else, it looks exactly the same. There are 20 different possible R groups, which are shown here. I will sometimes reference amino acids by their one-letter abbreviations. You don't need to worry about knowing all of them. I will tell you that F and Y seem to be ones I'll just like a lot. So I'll probably use F and Y as examples a lot. But what I want you to notice about the R groups, which are the things that differ from amino acid to amino acid, is that they vary quite a bit in terms of whether they have a charge, in particular whether they're basic or acidic, so they have a plus charge or minus charge, whether they're hydrophobic, and how big or bulky that side chain is. And so you can imagine, and you can think of all of these amino acids as having different shapes, which is dictated by this different R group. And so I'm going to make mention of both the backbone or the main chain atoms and R groups at different times today. At this point, I have to kind of stop talking about MHC as MHC and start to break us down into MHC class 1 and MHC class 2. Because MHC class 1 and MHC class 2 are kind of the first big place where MHC really varies. And they even start to vary in terms of the structure of the proteins. And so as we talk about the structure of the proteins, we're going to be thinking specifically first about the class 1 structure and then about the class 2 structure. And so here you can see kind of a compare and contrast from a different textbook of the MHC class 1 structure and the MHC class 2 structure. I'm going to kind of go through the details of class 1, and then I'm going to go through the details of class 2. But this is sort of the general compare and contrast first. And so the first molecule that we're going to talk about is the MHC class 1 molecule. I will admit MHC class 1 is the one I have spent considerably more of my life thinking about. And so usually in my head, I default to class 1 pretty often. So first, we're going to think about class 1. So what you can see here is that MHC class 1 has two different protein chains. One is shown in yellow, one is shown in green. The yellow one is the alpha chain, sometimes also known as the heavy chain. I put all the good chalk over here. One of the key things to know about MHC class 1 is that the alpha chain has a transmembrane domain. So our alpha chain actually goes through the membrane. It then has an immunoglobulin domain I was always taught that to draw immunoglobulin domains kind of like this. So whenever I draw this in a protein, it's an immunoglobulin domain. So it's got an immunoglobulin domain, and then it's got two other domains that look kind of like that. They're not quite immunoglobulin domains, so they're not drawn as perfect immunoglobulin domains. And these are known as the alpha 3, alpha 2, and alpha 1 domain. Another big piece to notice about MHC class 1 is that MHC class 1 also has a partner protein. And that partner protein is also an immunoglobulin domain. I have just drawn it poorly so that they're different sizes. They should be the same size. And this is called beta 2M, or beta 2 microglobulin. You can see that class 2 is going to have a slightly different structure. And so here you can see your textbook's view of MHC class 1. We can see the transmembrane domain of the alpha chain, then an immunoglobulin-like domain, and two other domains. And you can see the immunoglobulin-like domain, or the immunoglobulin domain of beta 2 microglobulin. Notice it is not connected to the heavy chain, so it is not covalently connected. It also does not have a transmembrane domain. So it's sort of just sitting here free. So if, say, I was asking you to be drawing one of these on an exam, those would be the things I would be looking for. And then in this little area at the top, we have our peptide binding groove. And so in the end, our peptide binds right there between the alpha 1 and alpha 2 domains. So this is, in fact, a complete MHC class 1 molecule. And so you can see that peptide binding groove, and in fact, you can also see that peptide binding groove here. Both the alpha 3 domain and beta 2 microglobulin are largely made of beta sheets, while the alpha 1 and alpha 2 that make up the peptide binding groove are largely alpha helices. And in fact, it's basically two alpha helices that are sitting like on a tray of beta pleated sheets, sort of up to present. I had a thought. Wonder where it went. Oh. So the other thing to note that I want to point out here is about beta 2 microglobulin. Beta 2 microglobulin is not encoded in the MHC region of the genome. That protein is encoded somewhere else. And it does not vary from person to person or from MHC to MHC. Basically, your body just makes a whole bunch of it. And all of your class 1s, whether they're A, B, or C, they all just use beta 2M. Like, you just make a ton of it. They all use it mix and matchy. One thing that's actually really important is that because beta 2M is non-covalently linked with the rest of this molecule, it actually can come on and off with an on-off rate. When it does that, it actually, the peptide comes out of the binding groove. This is not folded correctly unless you have all of these things. And so in reality, we actually think that what's happening is beta 2M is going on and off at some rate, and it's sort of like throwing and catching the peptide at some rate. So sometimes people talk about how MHC class 1 breathes. And so it does sort of have that movement to it. So there are a couple of really key things about the MHC class 1 binding cleft. So this is these are the images from your textbook of the MHC class 1 binding cleft. And so what you can see is that the MHC class 1 binding cleft has these alpha helices, one coming from the alpha 1 domain, one coming from the alpha 2 domain, and then kind of there are those beta pleated sheets making a little tray underneath. So right now, you are the T cell, and you are looking at the MHC. This is empty one. This is MHC with peptide in the middle. So you can kind of see MHC plus peptide. You can see what that MHC looks like. This is another view of the same thing, though with a space-filling model. And I actually find this one to be a really important one to show you. What I want you to notice, so the white is the alpha alpha 1 and alpha 2 domains. The white is the MHC, the red is the peptide. What you should notice is that these areas here on either side of the peptide are sort of pretty tight around that peptide. The peptide cannot be have a lot of variation in its length. You can't really have a peptide that's any longer or shorter here because those ends are completely closed. And so with MHC class 1, one of the things that is really important is that it can only bind peptides that are between 8 and 10 amino acids in length. And it's very strict. And when I say between 8 and 10, usually, like, if I'm talking about HLA A2, it binds 8. And if I'm talking about, like, A3, it might bind 9. And if some other one, it binds 10. So 8, 9, or 10 are the only choices. And this is all because these ends here of the peptide binding cleft are closed. So basically, these two alpha helices come together really tightly and close up that peptide binding cleft, so the peptide doesn't have a whole lot of flexibility in terms of its length. And so that's a key feature of the MHC class 1 peptide binding cleft. You can also see that the MHC class 1 peptide binding cleft is drawn here entirely in white because it's all made of the same protein. The other really key thing about MHC class 1 peptide binding cleft is if you look at the biochemical interactions between the peptide binding cleft and the peptide, those interactions are interactions between the binding cleft and the R group of the peptide. So with MHC class 1, we are seeing binding to specific R groups. What that means is that the MHC class 1 molecule cares which amino acids are in the peptide. It can't be any old peptide. It has to be a peptide with the right amino acids that can fit, that have the R groups to bind. And we, in fact, refer to those R groups on the peptide as the anchor residues. So there have to be some specific R groups, say circle R group or cone R group, that bind into specific pockets of that MHC class 1 binding groove because we are seeing specific interactions with the class 1 binding groove and the R groups here. And so we have these anchor residues, which are amino acids that have to be a particular residue. And so what people have realized with MHC class 1 epitopes is that people have actually taken different class 1s. So here's class 1 variant 1. You could imagine that as being A1 versus A2, or A1 versus B1, or whatever the heck you want them to be. I don't remember what they were in this real paper. But people basically just took all of the HLA-A1 from cells and did biochemistry to pull the peptides out of the peptide binding groove. And then they just sequenced the peptides. And what they found, for example, is that those peptides always had a G at position 2, and they always had a P at position 3. They always had an L or I at position 9. So they said, well, then the anchor residues for this number 1 are G at 2, P at 3, I or L at 9. And they did the same kind of thing. They basically pulled all the peptides out of another kind of MHC and sequenced them. And they found that they always have Y at position 2 and I or V at position 9. And so this one has different anchor residues. This experiment was actually done by 2 undergraduate students in Germany. They're not undergraduates anymore. This was a long time ago. But this was an undergraduate thesis project. Yes?
Do you want us to know, like, the location of the residues and what they are?
No, you need to know what anchor residues are, because for all of those 3,000 HLA-Bs, the anchor residues are different. And so you don't need to memorize 3,000 types of HLA-Bs, but you need to know what an anchor residue is. And so if I describe to you, or if I say, in this kind, the anchor residue is number 3Y, then you should know to look for number 3Y and like what that means. And so this is sort of another way of showing aspects of anchor residues. So for example, this type of MHC class 1 can bind any peptide that has L or M at position 2, V at 6, and V or L at 9, such as this example. While a different one binds anything with R at position 2 or R or K at position 9, such as this example. And this is based on things like the shape and the charge of those amino acids. And so that's how we have different MHC class 1s that can bind to all sorts of different peptides from many different microbes. One other thing I forgot to mention with the MHC class 1 binding cleft. So this is our MHC class 1 binding cleft. We usually have some fun shapes. That might allow us to have, say, a lysine and a tyrosine. That could be an example. We also actually have two other patches in the class 1 binding cleft. This patch here is all full of negative charges. This patch here is all full of positive charges. And that's because the end of this peptide is positive and the end of this and the other end is negative. So the N terminus is always positive. The C terminus is always negative. And so in fact, I told you before that the ends are closed, and we even have nice little charged patches to attract the charges and get everything to fit all nice and tight. So this is another kind of key feature of the class 1 binding cleft. So that's what we see with the class 1 binding cleft. There's another key thing to know about MHC class 1. Well, there are a bunch of key things, but another one that I'm going to tell you on the next slide, which is where MHC class 1 gets peptides from. So MHC class 1 and MHC class 2 vary in terms of where they get their peptides from. MHC class 1 gets all of its peptides from the cytoplasm of cells. So MHC class 1 specifically is presenting cytoplasmic peptides. So the peptides that get onto MHC class 1 are all from the cytoplasm. The most famous, or the easiest to understand, peptide protein in the cytoplasm is a protein that came from a virus that's trying to replicate in the cytoplasm. So if a virus was replicating in the cell, it's replicating in the cytoplasm. And so most famously, like if I come up with some neat little story on an exam and I'm like, and there was a virus, blah, 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 I want it to be class 1. Usually when I think of virus, I think virus and class 1 go together in most cases. Now, if you think about a cell that is infected with a virus, is there a way that we can go through and sort that cell and take out the virus parts and leave the rest of them intact? No, not really. So if this cell says, hello, look at my proteins, and the T cell recognizes a viral protein, is there anything the T cell can really do for that cell to make it healthy? No. Nope. And so cells that recognize MHC class 1 are the killer T cells. So the killer or cytotoxic T cells all recognize MHC class 1. And I will show that to you again in a couple of slides. In fact, I'm looking at my order of slides. I'm realizing that I officially am supposed to tell you that in a couple slides, but it's on this one too, so I'll tell it now. So there's one other kind of little bit of information I have to point out here. If you look at the affinity of the T cell receptor for MHC plus peptide, you find that the T cell receptor does not bind MHC plus peptide very tightly. It's a pretty weak binding. As a result, if we really want our T cell to interact with our infected cell, get some signals, do some stuff, the T cell receptor needs friends to help give it stronger binding. Because its binding by itself is weak. So we need to have some other proteins helping out. In the case of MHC class 1, that helper protein is called CD8. So MHC class 1 always goes with CD8. So you can see that this is our MHC class 1 plus peptide. You can see that the T cell receptor is binding to that MHC class 1 plus peptide. But you can also see that CD8 is binding sort of to this conserved part in the back of MHC class 1. That's a part that doesn't vary. So for every single MHC class 1 kind of looks the same right here, and CD8 can bind to all of them. So the friend in the case of MHC class 1, officially the co-receptor in the case of MHC class 1, is CD8. And so the type of T cell that recognizes MHC class 1 is called a CD8 T cell because it has CD8 on its surface. Yeah, Jamie?
Is there any sort of advantage to the T cell receptor having low affinity, or is it something that we don't understand why it does that?
I would say that we don't really understand why it does that, although this co-receptor coupling is pretty great. So I don't know if I will be teleological enough to say that it's so we could have this, but it's kind of a nice feature. Is my simple answer. I could give you a more complicated answer, but we're going to go with this as a simple answer. In fact, as I'm sitting here thinking, I thought of a much more complicated answer for like a few weeks from now. And so MHC class 1 plus peptide is the antigen for the CD8 positive T cell. So specifically when we talk about CD8 positive T cells, the antigen that we're seeing is MHC class 1 plus peptide, which is giving us peptides that came from the cytosol. We're turning on the CD8 T cell, which is also the killer kind. Because remember, we want to kill a thing that has bad peptides in the cytosol. So there's one last piece of info that I need to tell you about MHC class 1. And that is which cells have the ability to do MHC class 1 presentation. Which cells have MHC class 1 on their surface and have the ability to show off cytoplasmic peptides. So let's say for a second that this is usually a thing that happens with viruses, right? So if you were going to design this system, what cells would you want to be able to show off potential proteins in their cytoplasm? Yeah, Jamie?
Cells in the respiratory tract in the lungs.
Okay, cells in the respiratory tract in the lungs. Any other cells that you'd want to have the ability to do this? Yeah, Sebastian? Maybe cells in the mouth? Rishi? Nasal cells? Jamie?
Probably any cell that can get infected by a virus.
Any cell that can get infected by a virus. Can you think of any cell that can't get infected by a virus? There's one kind. What is it? Red blood cells, because red blood cells don't have nuclei. So in fact, if we look at which cells make the MHC class 1 protein and have it on their surface, the answer is all cells in your body except for red blood cells. Oftentimes we refer to this as all the nucleated cells, because the ones that don't have the nucleus, they don't have MHC class 1. So oftentimes we say MHC class 1 is on all of your nucleated cells. So right now, all of your cells except your red blood cells have 6 MHC class 1s on their surface. 3 from mom, 3 from dad. So go cells. All right, those are all of our facts about MHC class 1. now we've got to kind of do the parallel facts about MHC class 2. So we're moving to MHC class 2. Unsurprisingly, MHC class 2 is encoded in the MHC class 2 region of the genome. And we can see the images of the MHC class 2 protein here. So the MHC class 2 protein also has 2 protein chains. Those 2 protein chains are much more similar to each other compared to the alpha and the beta that we saw for class 1. One is called alpha, one is called beta, but they both have transmembrane domains. They both have immunoglobulin domains. They both have these kind of more unstructured domains. And so you can see the structure looks very different. If I was kind of looking for you to draw this, I'd want 2 transmembrane domains. I'd want to see, this and this covalently linked, this and this covalently linked, but these are not at the top, unlike what you see with MHC class 1. And the peptide binding groove is being made of this alpha 1 and beta 1, so parts of 2 different proteins coming together. Although you can see that still is a set of 2 alpha helices with a beta pleated sheet tray under it. One thing that is pretty cool about the MHC class 2 molecule, for any of you who are in biochem, is that when you have MHC class 2 plus a peptide in the peptide binding groove, so the peptide's not drawn here, but if there's a peptide drawn here, this complex of these 3 chains is incredibly stable. So while I said the class 1 kind of breathes a little bit, class 2 is incredibly stable. It is actually known as an SDS stable molecule. So you can boil this in SDS, which is usually what you do to unfold proteins, and this whole complex of proteins does not come apart. It's really tightly put together, and that's going to become important next week. If we look at the MHC class 2 peptide binding cleft, it looks a little bit different than the class 1 binding cleft. So first, you can notice from this space fill model that the class 2 binding cleft is made of 2 different proteins, the alpha and the beta. So there's white and there's blue. But what you can also notice is that the peptide in orange kind of goes further across the whole thing. And in fact, the ends of the peptide binding cleft are open here. So the peptide can actually vary quite a bit in length. And you can see that here as well. So it's not tightly together. You don't have these little charged regions because basically the ends here are open. And the ends of the peptide can flop out the sides of it. And every immunology textbook I know of makes a comparison here to a hot dog bun. And about half of them then show you a picture of a hot dog in a hot dog bun. But I was pretty sure you knew what that looked like, so I did not need to put one up here. But so class 2 is the one that has the open ends, and so you can have the peptide hanging out the ends. So with class 1, we're pretty strict about how long the peptide is. With class 2, we're not as strict. Often I hear people say, sort of the idea is like 10 to 20-ish amino acids. But I've heard of way longer ones. So it's really a lot of variation that's possible with the peptides binding class 2. In addition, if we look at MHC class 2's interactions with the peptide that is being presented, the interactions that we see are with the main chain atoms, that NCC, NCC, NCC in the middle. And that NCC, NCC in the middle isn't different from amino acid to amino acid. So we're getting kind of a much less specific binding to the peptide here because class 2 is interacting with those main chain atoms. Sometimes people try to say there are anchor residues for class 2, but they're really not good. I do not believe that really we have much in the way of anchor residues. So we can look at this. Oh, we got all these peptides out of the same MHC class 1 molecule. Well, This one has an L at position 5, and this one has an F at 4, and an I at 5, and an I at 4, and an F at 4, and an I at 7, and this is an L at 7. Like, it's not at all the same kind of very strict, always this amino acid at this number pattern, because the peptides are of different lengths. So where one starts is different. You can see there's a lot more variation in terms of which amino acids are going to be in which places. You really don't see the kind of strict epitopes on class 2 that you see with class 1. So MHC class 2 specifically picks up its peptides from the endocytic pathway. We'll see a lot more of the endocytic pathway next week. But basically, these are membrane-bound compartments inside of cells. So this is different in terms of the source of peptides. MHC class 2 does not interact with CD8. Instead, MHC class 2 interacts with CD4. So you can see it's the same kind of interaction, but it's a different protein. In this case, it's CD4 instead of CD8. And as a result, MHC class 2 plus peptide is the antigen for CD4 positive T cells. Those are the helper ones, the ones that make a lot of cytokines. And so I've got 3 slides left. 2 of them are summary for your studying purposes, so I don't really have to worry about them. So I just have to do this one. And this one is what cells express MHC class 2. That compartment where I showed you we get our MHC class 2 peptides, and we'll see a lot more of this next week, is a compartment that's often involved in phagocytosis. And so by and large, the cells that are doing class 2 presentation are phagocytic cells. In fact, they are really known as our professional antigen-presenting cells. And you have only 3 cell types in your body that are professional antigen-presenting cells and that can present on class 2. And those are dendritic cells, macrophages, and B cells. So as far as you're concerned, only 3 cells of your body do class 2 presentation, which is dendritic cells, macrophages, and B cells. And so all of the cells in your body do not have the class 2s on the surface. These cells have class 1s and class 2s. It's not like they have only class 2. They have both, because they're still nucleated cells. I will also point out here, there are some cells of the thymus that also have class 2 that are important in T cell development. So we will see those when we do T cell development. But outside of T cell development, only the 3 professional antigen-presenting cells have class 2 on their surface. And if you would like compare and contrast of class 1 and class 2, here are 2 slides with summaries of all the info. So we are going to see kind of the cell biology of how class 1 gets its peptides on Wednesday, and then how class 2 gets its peptides on Friday. Have a great fall break, and I will see you guys next week.